Today we are going to learn in vivo toxicity studies using zebrafish embryo. In today's lecture we are going to learn various experimental models and advantages of zebrafish as an animal model. In today's lecture we are also going to learn how we can use zebrafish for understanding the toxicity of nanomaterials both theoretically as well as practically through demonstration. Let us see some of the current in vivo and in vitro models in human research. The in vitro model system we already learnt in some of the previous lectures. So, we can make organoids, we can have spheroids and tumoroids for understanding the toxicity of materials, but the major problem is low clinical relevance. And the advantages are like it has a high availability for screening and reduced cost and high throughput. So, these are the various advantages of this in vitro model, but the drawback is failure to replicate the native conditions. Whereas these in vivo models, it provides a various degree of physiological similarities to the humans, and also there are various genetic tools are available for genetically modifying these animals, but the major challenge is very expensive to maintain and limited availability, and also failure to predict the human response, and also we have the ethical and societal challenges when we are using this kind of animal models. Let us see some of the common in vivo laboratory models. We have this fruit fly and zebrafish and mouse model. So, most of the lab they use this kind of in vivo models for their research work, and initially we start with the in vitro model to understand the toxicity of the particular materials, and because the cell culture this in vitro model is very easy to work and low cost to maintain, and also the short experimental cycle. So, these are the advantages of this in vitro model system, but as I told earlier these are highly controlled and simplified systems. So, there is a poor correlation with the in vivo mechanism. So, that is why we have to move from in vitro to the in vivo model. But in the in vivo model, as I told earlier, there are a lot of challenges. We have to select the right in vivo model for understanding our materials toxicity. We can use the fruit fly. It is very easy to work because it has a short generation time and it is a very low cost of maintenance. But it has a limitation like it is genetically distant from the humans, and it is a relatively simple anatomy, and it has a no adaptive immune system. We can use the mouse. The mouse has a lot of advantages like it has a complex behavior and organ homologs to humans and genetic similarity to humans. But the limitations I told earlier like these are very expensive to maintain, and the experimental cycle is long, and ethical constraint. Whenever we use animal like mouse model, we have to take the ethical committee approval, animal ethics committee approval, and it takes some time to get the approval, as well as to maintain these animals also expensive. So, that is why like the zebrafish is an alternate to an animal model, and it is a very simple system. Advantages like it has a high reproductive rate and the development is external, and again it is has also has the genetic similarity to the humans, and the limitations like again it has the moderate flexibility and moderate predictivity, and also moderate translational value. Let us see why zebrafish is a human model and what are the advantages of this zebrafish. Zebrafish is a common animal model for scientific research because of their many similarities to humans and easy to maintain as well as easy to genetically modify. And if we see that humans and zebrafish share almost 70% of their genes, and these zebrafish embryos are transparent. So, it will be very useful for the researchers to observe their growth under microscope and track the effects of drug or genetic modification. We can add a drug, we can add nanoparticle, and we can monitor under the microscope. And also these zebrafish are very small, so it can be easy to keep in big groupings. Also this zebrafish may breed all year round and create hundreds of embryos per week, and they also grow very fast. So, these are the advantages of this using zebrafish as a human model. Let us see why zebrafish for the toxicology studies, because as I told earlier it is easy to propagate and monitor the developmental stage because the zebrafish embryos are transparent in nature, and here this DPF means it is a days post fertilization and HPF means hours post fertilization. And additionally this zebrafish also has a 70% genetic homology with the human species, as I told earlier. So, this zebrafish can be a very good model for understanding the various human diseases. From this slide we can clearly understand the conserved organ system between the zebrafish and humans. So, that is why the zebrafish can be a very good alternate animal model for understanding the various human diseases. Let us see what are the various experimental studies we can perform using zebrafish model. The first one is behavioral studies. For example, we can use a zebrafish model for understanding the anxiety, and in pharmacology we can use the zebrafish model for understanding the diet or drug induced obesity, and we can also have the zebrafish as a model system for understanding the type 2 diabetes as well as non-alcoholic fatty liver diseases and other intestinal diseases. In toxicology, we can do various toxicity studies including neurotoxicity, genotoxicity, and also nephrotoxicity. So, that is why this zebrafish is a very good model system for understanding the toxicity of nanomaterials. Let us see how we can use a zebrafish model in aquatic ecotoxicology. So, we can use the zebrafish model for understanding the heavy metal pollutant toxicity, and if there is a heavy metal contamination in the water and that leads to neural pathways or block, and also the cell death apoptosis will be increased and that leads to myocardial hypertrophy also. And if there is a more amount of organic pollutants in the water, that will decrease the activity of zebrafish, and also there will be a skeletal abnormality. And we can also use the zebrafish for understanding the nanoparticle toxicity in the water. So, for example, if there is a nanoparticle toxicity that leads to embryonic malformation or death of the zebrafish, or it leads to impaired immune function as well as the various inflammatory reactions. Let us see how we can study the toxicity of silver nanoparticles using the zebrafish embryos. So, the zebrafish embryos will be placed in a 96 well plate. So, we have to keep the zebrafish embryo in the 96 well plate, then add various concentration of silver nanoparticle. So, when you add various concentration of nanoparticle and after incubation, you can see the zebrafish embryo under the microscope, and at low concentration you may observe this kind of normal embryo. That means that particular concentration is safe for use. So, that is there is no change in the morphology and the embryo is normal, and at different concentration you can observe the embryo is malformed. If you are observing this kind of malformed embryo, that means that concentration is not safe to use for further studies. And if you are using high concentration of silver nanoparticle, you may observe this kind of dead embryo. The toxicity of silver nanoparticles can also be studied by observing the heart rate of zebrafish embryo. With respect to the increasing concentration of silver nanoparticles, the heart rate is decreasing and the mortality rate is increasing. Let us see another example for understanding the toxicity of nanomaterials using zebrafish embryo, and here we are going to learn about nanoceria. This nanoceria nanomaterial has the antioxidant activity. It mimics like the enzyme that is SOD, that is a superoxide dismutase enzyme, and it removes the reactive oxygen species. And here we are going to use this nanoceria, which is encapsulated in an albumin nanoparticle, and we are going to understand its production efficiency, how it is going to protect the zebrafish embryo in presence of hydrogen peroxide. So, for that we added this nanoceria to the zebrafish embryo, and here we did not add any nanoceria, and these are the different concentration of nanoceria added to the zebrafish embryo. Then we added these hydrogen peroxide. We can see here when there is no nanoceria, the reactive oxygen species is more, and in case if you are using the different concentration of nanoceria, you can see here the reactive oxygen species is going down, because this nanoceria is removing these ROS by its antioxidant enzyme like activity. And for this assay we use DCFS-DA assay, which you already know the principle of this assay. Most of you know that in DCFS-DA assay, if we have more ROS, then the fluorescence will be high. You can see when you are using nanoceria alone, there is no fluorescence, and also in the control where we did not add any hydrogen peroxide, we did not add any nanoceria, there is no fluorescence. But when you are treating only with the hydrogen peroxide, you can see high fluorescence. So, that is due to the high amount of production of ROS. But when you are incubating this zebrafish embryo with the nanoceria, you can see here the ROS is suppressed. So, that is why you can see here there is no fluorescence. That means the ROS is suppressed by this nanoceria, which has the enzyme like activity, and it is removing all the ROS. And we can also understand the toxicity of this nanoceria, and when you are using the nanoceria of different concentration, you can see here. So, there is no toxicity, it is almost like 100%. So, the survival rate is almost 100%. And when you are using this nanoceria in presence of hydrogen peroxide, you can also see here when you are not using the nanoceria, okay, so the survival rate is going down. But when you are using this nanoceria, with respect to concentration, the survival rate is also increasing. That means, so the nanoceria is protecting the zebrafish embryo from the reactive oxygen species. So, from this slide we understood how the nanoceria can increase the survival rate of zebrafish embryo and also the protective effect of nanoceria against the hydrogen peroxide induced embryotoxicity in zebrafish. Let us learn how to identify the male and female zebrafish. You can see here this is the male zebrafish, and male zebrafish can be identified by the small and slim body size, as well as we can see the bright color when compared to the female, and also it has a long fin. So, these are the identification marks for the male fish, and in case of female fish you can see here it has a rounder body when compared to the male, and also it has a large size, and it has a dull color when compared to the male, and it has a small fin when compared to the male. So, based on this we can identify which is male and which is female fish. To get the embryo for bioimaging experiment, we have to keep the fish for breeding. Before we keep it for breeding, as per the OECD guidelines, test number 236, that is a fish embryo acute toxicity test, we have to keep 2 male and 1 female fish in the spawning unit, and this is the spawning unit, and this is a recommended male to female ratio for zebrafish breeding, because this ratio increases the chances of successful fertilization. And here in the spawning unit, the male and female fish are separated by a transparent barrier for overnight. Then the next day, remove the transparent barrier, allow the fish to breed. After breeding, the fish lays the eggs, which can be collected and counted for the imaging studies. And we have to select the healthy embryo for our studies. So, how to select the healthy embryos? If the egg is transparent and shiny, it is a healthy developing embryo. Instead, if you observe white opaque egg, then it is a unfertilized egg or it contains a dead embryo. Once you select the correct embryo, count it, then add the carbon darts and incubate it. After incubation, you can observe the carbon darts labeled fluorescent embryo using fluorescence microscope. From this picture, we can understand that after incubating zebrafish embryos with carbon darts, you can observe the fluorescence. And this is a bright field image, and this is a fluorescent image, and this is the merged image. And from this image, you can clearly understand that there is no toxicity of carbon darts compared to the control. And also we can understand the bioimaging applications of carbon darts from this image. Let us see the maintenance, breeding and typical conditions for zebrafish embryo acute toxicity test as per the OECD guidelines. We have to feed the zebrafish 3 to 5 times daily with the dry flake food. Additionally, we also have to give brain shrimp, that is Artemia, because feeding live food provides a source of environmental enrichment, and to guarantee for optimal water quality, the excess food and feces should be removed approximately 1 hour after the feeding. The other parameters such as water temperature and water quality has to be maintained properly. Also, we have to maintain the proper illumination, and we have to follow the photo period, that is 12 hours lights on and 12 hours off. And as I told earlier, we have to follow the recommended male to female ratio for breeding. Based on the egg structure and appearance, we can select the healthy developing embryo. As I told earlier, if the egg is transparent and shiny, it is a healthy developing embryo. Instead, if you are observing a white opaque egg, then it is an unfertilized egg or it contains a dead embryo. And the spawning rate, so that mainly depends on the strain. Usually, the single mature female spawns at least 50 to 80 eggs per day. I hope you got the overall idea about how we can use the zebrafish embryo for understanding the toxicity of nanomaterials. Let us go to lab and learn this more in detail. In this experiment, we will learn how to do bioimaging of carbon darts by using zebrafish embryos. So, to begin the experiment, we require zebrafish embryos. First, we need to separate the male and female fishes in a 2:1 ratio using transparent glass barrier. Once this is done, switch off the light and leave them overnight. Now we have switched on the light. The next step is to place the chamber at a slanted angle and remove the transparent glass barrier placed between the male and female fish. Leave the setup undisturbed for 20 to 30 minutes. The fish may seem to be fighting or chasing each other. These are the signs of spawning. The female fish releases eggs into the water, which are then fertilized by sperm from the male fish. Here we can observe the eggs in the bottom of the chamber. Now we will remove the fish and place them in another chamber to collect and clean the embryo. The separated fish can be further separated into female and males, fed, and it will be ready for breeding in next few days. Next, we will observe the embryos under the microscope to identify and separate the fertilized embryos. To clean them, we will use E3 media. It is important to handle the embryos carefully to avoid damage. Now we will view the embryos under the stereozoom microscope. We will see the embryos on this large screen. First, we will open the software on the computer. Then we will place the plate under the microscope. Switch on the background light. And change the light mode to bright field. Once the embryo is clearly visible in the eyepiece, adjust the knob from bright field to dark mode so that we can see the embryo in the monitor. Switch to live mode, adjust the settings. and capture the image for reference. We have separated the fertilized embryos. Now we require the test material, that is carbon darts. We will prepare the dilution of carbon darts to achieve the desired concentration. Once we have prepared the desired dilution, we will treat the embryos. First, we will place the exact count of embryos in the 12-well plate. Remove the media. And for the control, we will add the E3 media. For the treatment, we will add carbon darts. Incubate it for at least 2 hours. Now we will place the control embryos on a slide. For the carbon dust treated embryos, we will wash them using E3 media and then place them on the slide. We will now view these slides under stereosome microscope for bioimaging. First, we will observe the control embryo. For bright field, we will follow the same procedure as I told earlier. To observe under the GFP filter, press the GFP button and switch it on. Then turn off the background light. As you can see, there is no fluorescence in the control embryo and it is not clearly visible compared to the bright field. Now we will place the carbon dioxide treated embryo. First, we will observe it under the bright field. You can see the embryo containing carbon dots. Next, we will switch on the GFP filter, then turn off the background light. Now we can observe the green fluorescence in the embryo, confirming that the carbon dots have entered the embryo, demonstrating their bioimaging property. Now let's compare the carbon dioxide treated embryo with the control embryo in the bright field. Finally, we will save all the images. In this lab session, we have learnt how to use zebrafish embryos to study the bioimaging properties of carbon dots. As a summary, in today's lecture, we learnt about various experimental models used in human research, and we also learned how we can use the zebrafish as an alternative toxicological model. Through practical demonstration, we also learned the bioimaging experiment using zebrafish embryo. Thank you for your kind attention. All the very best for your exams.